Bismillahir Rahmanir Rahim. Assalamu alaikum. I am Professor Adam Ahmed Adam. This immunology lecture is about acquired immunity. This immunity is a specific immunity. That is to say, each component is specified to a specific antigen. It is gained by the body after antigenic stimulation to the immune system. It is of two types. One, humoral immunity, which is mediated by antibodies. Two, cell-mediated immunity, and this is mediated by cells. Now we come to humoral immunity, the one which is mediated by antibodies or immunoglobulins. I would like to draw your attention here that the antibody is the same as the immunoglobulin. Antibodies are glycoproteins which are produced by plasma cells after antigenic stimulation to the B cells. This drawing will make the information clear for you. Now you see on the upper left hand side, the specific antigen stimulates its specific B cell, and this B cell is going to be activated and it divides into many cells, and these cells are going to be grouped into two groups. One group, the memory cells, lower down here, the green arrow. These memory cells are long-lived lymphocytes that keep immunological information about this antigen. So if this antigen comes in the common occasions, it will be detected in a short time and the immune system mounts an immune response against it. The other group, the plasma cells, look here on your right hand side. These cells, their function is to produce and secrete antibodies. There are 5 classes of antibodies. They are so named according to the name of the heavy chain. This is the basic unit of an immunoglobulin. Each class of immunoglobulin is composed of at least one basic unit. Some of them have more than one basic unit. Each basic unit is composed of two identical heavy chains and two identical light chains linked together by disulfide bonds. In an immunoglobulin or antibody, the light chain can be kappa or lambda. It doesn't make any difference. Then look at the upper part of the drawing, the amino terminals. There are two arms. Each arm is an antigen binding site. So the basic unit of an immunoglobulin can bind to antigens. At the very end of each arm, there is a darkly shaded area. This is known as variable area. So there are two variable areas, and the variable areas are composed of amino acids that change according to the specificity of the immunoglobulin or the antibody. So the variable areas determine the specificity of the antibody. Lower down here, you see the carboxyl terminal. There is a receptor. This is a non-specific receptor. According to the type of the heavy chain, we can find the classes of the immunoglobulins. We start with the first class, IgM. Here the heavy chain is mu. It is a pentamer, that is to say composed of 5 basic units. It constitutes about 10% of the total immunoglobulins in the blood. It has got the ability to fix the complement. We are going to talk about the complement system in a separate lecture. It has a molecular weight of 900 kilodaltons. It is the heaviest immunoglobulin. It is the first immunoglobulin that appears in primary immune response, that is to say the immune response for the first time. It mediates agglutination. The other class, IgG, its heavy chain is gamma. It is a monomer. It is composed of 1 basic unit. It appears in the secondary immune response. It constitutes about 75% of immunoglobulin in the blood. It is the most abundant immunoglobulin. It has a molecular weight of 150 kilodaltons. It is the lightest immunoglobulin. It crosses the placenta from the pregnant woman to her baby in the uterus. It is the only immunoglobulin that crosses the placenta. It fixes the complement. It opsonizes the antigens to facilitate phagocytosis by the phagocytic cells. It mediates agglutination, so it is an agglutinator. It has 4 subclasses from 1 to 4. IgA is heavy chain is alpha. It is about 15% of the total immunoglobulins in the blood. It protects both the mucosal surfaces. It is found as a dimer of 2 basic units in body secretions. It has a molecular weight. This dimer has a molecular weight of 400 kilodaltons, but it is found as a monomer in serum with a molecular weight of 160 kilodaltons. IgE, its heavy chain is epsilon. It is a monomer. It has a molecular weight of 190 kilodaltons. Its concentration increases in type 1 hypersensitivity reaction and parasitic infections. It mediates inflammation. IgD, its heavy chain is delta. It is a monomer with a molecular weight of 170 kilodaltons. It is found on the surface of B lymphocyte. It is part of the antigen recognition signaling mechanism on Virgin B cells. Virgin B cells, the non-stimulated cells. The protective mechanisms of antibodies. Number 1, direct action or neutralization by IgA. By this, the IgA nullifies or abolishes the effect of the uh, pathogen or the antigen. 2. Agglutination. Antigens clumping. The antigens are clumped and are not allowed to spread, and this is done by IgM mainly and to a lesser extent by IgG. 3. Opsonization of antigens facilitating phagocytosis, and this is done by IgG. 4. Inflammation by IgE. 5. Complement fixation by IgM and IgG. Now we come to the cell-mediated immunity or cell-mediated immune response. Its function is to protect the body against intracellular antigens. These antigens may be pathogens like bacteria, viruses, and so on, tumors, which are abnormal growth or neoplasm. This immunity prevents the development of tumors. Graft or transplant antigens rejection. And also I'd like to emphasize that killing of virally infected cells. So to start with, look at number 1. The antigen of these substances will be presented by the antigen presenting cells, and the fragments are displayed or put in the MHC class 1 here. Look at the blue area in the antigen presenting cell. Then the helper 1T cell will come to detect the antigen fragments here, and then it becomes activated and proliferates into many cells. One group will be the memory cells, and the other group of cells, these cells are going to secrete cytokines. Examples of cytokines: interleukin 2 and interferon gamma, and there are other cytokines. The action of these cytokines here, look at the three on the left hand side. Activation of the macrophages to phagocytose the invading pathogens. So the macrophages are going to be aggressively phagocytic. Then look at the 3 on your right hand side. These cytokines stimulate the cytotoxic T cells to kill pathogens, tumor antigens, and transplant antigens, and especially also killing of the virally infected cells. By this, the lecture is over. I hope that it is understandable, and I wish you all the best.